Yeah, so good afternoon. Yeah, it's sunny now as compared to last few weeks when it used to get dark. So I think I can wish you good afternoon. It's not evening yet. Um, so the days are getting bigger, and but I still see the snow all over the street. So I heard that Sunday there is going to be one more load of snow. So take care. And let's get into the slides. So I had uh, I was here at Recombinant DNA Technology. Uh, so I will start from that point. And before I start, I see a student who just says Zoom user. For the sake of attendance, this is a synchronous setting. Could you let me know your name? Person who just got in and his or her, it shows up as Zoom user. So anyway, so I was talking about so I see somebody as Claudia Valles. Who is this? Because I don't have anyone by the name of Claudia Valles.
It's Wavelength.
Okay, great. Thank you. Thank you. So, we stopped the class around this slide, and on this slide, I was trying to emphasize that bacteria is a lab in itself, because in a lab, in a typical lab, what do? You do an experiment, and you have tools. So, bacteria has those tools that makes the experiment, the strategy for recombinant DNA technology feasible. And what are some of the tools? One is restriction enzyme. The second is plasmid. So restriction enzyme is an enzyme that can help to cut the DNA, put a nick in the plasmid, and then the gene of interest that we often talked that this will be insulin can be inserted. So we'll go into this insertion part and restriction digestion part later quickly. And then this plasmid becomes recombinant, right? It has a bacterial origin because plasmid is extrachromosomal DNA that's present inside bacterial cell. And it has a human insulin gene, for example. So human plus bacteria, it becomes a fusion or recombinant DNA. So that's why, classically, this is known as recombinant DNA technology or genetic engineering. And this recombinant plasmid is again reintroduced into the bacterial cell. And this plasmid has all the element to express this insulin gene into insulin protein, because this plasmid will utilize the same system of transcription and translation that a main chromosome will do. And then insulin on any protein coded by this insert from human gene will express, and then this can be purified, extracted from the bacterial cell to make a quantity of insulin. So I use the word restriction enzyme. So what is restriction enzyme? So so let's say let's first break this into part. What is enzyme? So enzyme is a catalytic or a catalytic agent that can increase uh, the rate of reaction, right? So now here we are talking about restriction enzyme, right? So what is restriction enzyme? Right? So first, let's talk about a special category of enzyme called as nucleases. Nucleases are those enzymes that can cut nucleic acid cut nucleic acid into pieces. We'll go into details later. So it will cut nucleic acid into details. Now uh, there can be two kinds of nucleases, right? Number one, nucleases that act on DNA, they're called as DNAs, and the nucleases that is called as RNAs. So as we have two types of nucleic acid, DNA and RNA, there are two types of nucleases also. The ones that act on DNAs is called as DNAs. One that acts on RNA called as RNases. Now, further, you can, biology is very vast. You can divide biological organisms or living organism into many ways. For example, you can divide the whole living organism by the presence of chloroplast or no chloroplast, right? So things that have chloroplast becomes plant, things that don't have chloroplast becomes, let's say, animal kingdom or something else. You can also divide biological organisms or living organisms depending upon presence or absence of nucleus. So those organisms that have nucleus are called as eukaryotes, and which don't have, they're called as prokaryotes. You can also divide on the basis of number of cells. All the organisms, whether prokaryote or eukaryotes, if they are made up of single cell, it is called as unicellular. If they are made up of multiple cells, then they're multicellular. So various ways, depends what kind of lens you have or magnifying lens you have. You can classify biological organisms or living organism into many ways. So likewise, you can also classify DNAs or nucleases into two ways. So first is called as endonucleases, and the second is called as exonucleases. So nucleases, right? Endo and exonucleases, right? I'm sorry, sorry for this typo. Sometimes when you write exo and nucleases, right? Exonucleases. Right? So now what's the difference? So let's say if this is a piece of nucleic acid, let's say DNA. So the exonucleases, they will act on extreme, right? They will, keep chewing the DNA or cutting the DNA from the extremes. But the endonucleases, they can act in the center or in the middle. So these sites are acted by endonucleases. So exo will act on ends, endo will act in the middle. Now, these restriction enzymes, the full form of restriction enzyme, they're called as restriction endonucleases because they can act in the middle. Now, why they're called as restriction enzyme? They're called as restriction enzyme because what happens? What's the favorite enemy of bacteria? The favorite enemy of bacteria is bacteriophages, right? So bacteriophages. these bacteriophagia, they can attack bacteria. And within this bacteriophage, there are DNA. So this DNA within bacteria, within bacteriophage, gets into the bacterial body. And then bacteriophage tries to take advantage of bacterial transcription and translation machinery, and then they proliferate, they become, and bacteria can be killed in this process. Now, some bacteria have the process to fight the attacking bacteriophage or protect itself. Just like we have immunity to many things, bacteria also have some kind of immunity. In our situation, the immunity comes from T cells, B cell, even let's say the tears in our eyes, it works as an innate immune tool, because if something gets into our eyes or nose, the mucus, the tears in the eyes can try to take care of our eye and take care of those dust or those little bit of insect if it comes. So in bacteria also, they have immunity system. And what's the immunity system? Is that they have sites within the bacteria which are methylated. Methylated means they have methylated DNA, means either adenine or cytosine, they can be methylated by. So methylation is addition of a methyl moiety. That is CH3. So bacteria has system which can prevent the bacteriophage to attack the bacteria. Now, how does it happen? So bacteria has enzymes. Bacteria has enzymes, right? This bacteria has enzymes which is called a restriction enzymes. So the moment when bacteriophage DNA enters into the bacteria, these enzymes, restriction enzymes, can chop this DNA into pieces. Now you can ask me a question that you're saying that, okay, restriction enzymes can cut the bacteriophage DNA, but why can't it cut bacterial DNA itself? I hope you're getting what I'm asking you the question. That restriction enzymes which are present in bacterial body can restrict bacteriophage infection. The same enzyme, why don't they cut the DNA into pieces? So the reason is that the site where restriction enzymes act, they are methylated. Now, what do I mean sites? So restriction enzymes are, so one of the property of enzyme is that they are very, very specific to the substrate. Just like you have a lock, and a lock can be opened by a specific key. So enzymes are also very specific. They act on particular DNA. So in case of restriction enzyme, for example, let's say there's a restriction enzyme in bacteria called as E. coli, and this has a restriction enzyme called as EcoR1, and they act at a very specific substrate. That substrate is this sequence. So I mentioned to you that enzymes are very specific. And for example, E. coli has a restriction enzyme, EcoR1. This EcoR1 acts at a specific sequence called as GATTC. Now, specificity will further come from this, that this enzyme will act here between G and A. And when I'm using the word, it acts at a specific site. So it will restriction enzymes EcoR1, they will need this sequence of nucleotide for them to act, right? And what do I mean act? Meaning that at this point of time, at this point, the DNA will break into two. And these enzymes, they specifically act at phosphodiester bond. So the phosphodiester bond between the G and A here will be cleaved. Now, one thing more interesting. So let's say I here just showed you that this is a single strand, right? But if you look at double stranded, right? A DNA is double stranded, GATTC. So, or it will be CTTAAG, right? So when you are reading this sequence from this side, and then again reading this sequence from this side, they read the same, right? So, and what are the sites the enzyme will act? Between G and A, between G and A. So such, for example, that such like words like madam, right? It can read in a similar way from either side, right? It will read from the sequence that will be same. Sequence will be same. Such sequences are called as palindromes, palindromic, or they're called as palindromic sequence, right? Which can read from both the ends. So, restriction enzymes are the enzymes which cut the DNA somewhere in the middle. The specificity of this enzyme comes from the sequence. This enzyme act at phosphodiester bond, and mainly those sites which are palindromic in nature. Right? Now, this is the background about restriction enzymes. Now, depending upon, so one way to study is what I encourage students, especially. There have been few students in my class who went to medical school or went to, in this class, who went to PhD program or DO program. So they had to take various kinds of tests. So I always sort of take this opportunity to suggest one of the way of studying is to study comparatively and quickly. So restriction enzymes can be classified on the basis of whether they need ATP, whether they need magnesium ions, whether they need manganese ions, or whether they have nuclease activities, or they also have methylase activities. So there are many ways a restriction enzyme can have, under many circumstances, restriction enzymes can work. Depending upon how they work, there are three classes of restriction enzymes. Class 1, class 2, and class 3 enzymes, right? Now, if you have such kind of chart in front of you, it will take you a few seconds to see what is what, as compared to going back and forth to the or reading the slides, right? So let's see what is the type 1 enzyme. So it has type 1 has both methylase and nuclease activities, and it requires both ATP and magnesium ion. So let's go back to our chart. So class 1 enzyme, it has both nuclease and methylase, and it requires manganese and magnesium and ATP. Class 2 enzymes, they are mainly nucleases, right? They don't have methylase activity. And let's just visit what's 2. So 2 do not require ATP. Magnesium or manganese is required. Methylase and nuclease are not in one enzyme. So mostly they are nuclease. And the reason we are studying this restriction enzyme to learn more about cloning. So this is the class 2 enzyme that is mainly used for cloning. So they have nuclease activity, and they don't require ATP, but they require magnesium, and some enzyme can require manganese. So this is the difference between the class 1 and the class 2 enzymes, right? So now let's look at class 3 enzyme. So class 3 enzymes do not require, class 3 enzyme, they have only methylase, do not require ATP, contains both methyl, so let's read systematically, contain both methylase and nuclease activity in 2 subunit enzyme. So it has so again, it has, this enzyme also has this has nuclease and methylase. And let me just go back and read here. They do not require ATP, but they do not require ATP, but could require magnesium or manganese. So what I was suggesting, that you can read systematically and comparatively, right? For if you're taking any exams, right, which requires you to have things memorized or in your system very quickly and efficiently. So you can see that ATP is required by only class 1 enzyme, right? Magnesium, or this part, all 3 enzymes need this cofactor. So magnesium is very, very important, right? All the 3 have nuclease activities. So all the 3 enzymes have nuclease activity. All the 3 enzymes require magnesium. Class 2 doesn't have methylase activity. So try to develop an approach like this, and this will help you to read systematically and save your time in terms of if you're preparing for exams which are of value. There's a cutoff value, for example, dental exam or MCAT, stuff like that. So now let's look at this picture. So here, if you look at the chemical level, right? If you look at the chemistry level, right? Yes, class 2 enzymes have nuclease. Yeah, they're not in one enzyme, right? But they have nuclease activity. So let's look at more carefully the chemical thing. So the enzyme, the restriction enzyme, they act at the phosphodiester bond between sugar. What sugar? Ribose or deoxyribose. So many students, they get confused that restriction enzymes break hydrogen bond. No, they break phosphodiester bond, okay, between phosphate and ribose or deoxyribose molecule. Now, when the restriction enzymes act here, so you have to imagine what is the bond between sugar and phosphate. I sort of talked about this. So this is OH, here hydroxyl group. This will give rise to hydroxyl group. When this bond is cut between sugar and phosphate, this gives rise to hydroxyl group and the phosphate group. And this becomes the site where cloning can be performed. So let's look at a different kind of picture. So here, the phosphate and the sugar bond is broken and it generates OH on the sugar and the phosphate. Now here I would like to add one more category. That's that. So let's imagine that this is the double-stranded DNA. So there are two kinds of restriction enzyme. One restriction enzyme acts symmetrically, and this is asymmetrical, two kinds. So symmetric is when enzyme cuts both the strands at same point. This is symmetric. But when an enzyme cuts at this place and at this place, so this is asymmetric bond. So let's take an example of symmetric and asymmetric. So see here, EcoR1, it acts at the site GAA-TTC. So the bond it is cutting here is between G and A, as I mentioned to you, but they are not at the same point in both the strands. There are different points here and here between G and A. So this is asymmetric. So EcoR1 is an asymmetric enzyme. But in contrast, if you see an enzyme called as PVU2, they are cutting at the same spot in both the strands, same spot. So this is symmetric. This is also called as blunt end. So depending upon how the cut by restriction enzyme is done, or the activity, if it is a different point at different location, then it is asymmetric. If it is at the same, then it is blunt end, right? So that's about restriction enzyme. Now let's look at some of the properties of restriction enzyme, right? So what you see here is a picture where you see here 8500, 5000, 3000. Then you see here 9500, 6000, 1000, and some. So if EcoR1 is digesting a DNA, it cuts at three parts. It gives rise to three fragments: 8500, 5000, and 300. When BAM-H1, 9500, 6100. So same DNA, when it is cut by two different enzymes, EcoR1 and BAMH1, it gives rise to three fragments, this one and this type. And when both of them cut together, it is giving a different pattern. So single, this pattern, enzyme A, enzyme B, enzyme A plus B, it gives different pattern. Now this has been utilized. This is called as restriction fragment length polymorphism. So this technique is one of the starting technique that was used for characterizing genetic characterization of bacteria or viruses or even forensic. If there is a suit like paternity suit. So now we have sequencing and nanotechnology and so forth. But in 20, 30 years back, restriction enzyme was a tool to identify many things. Why? Because they are enzymes that act at a specific target. So EcoR1 would act at a sequence of GAA-TTC, BAMH1 at GGA-TTC, and they will have their own unique pattern. And so let's say this is one of the pattern of an individual, and we are looking for a crime suspect. So in crime suspect, what happens? You're looking at the site, who is the victim and who is the convict or who is the criminal. So if the criminal DNA has lived there, is there, then you can match with the suspect. So you have 5 different suspects and you have 5 different patterns, and somehow the criminal has left a piece of his hair follicle or skin there. So restriction enzymes can exactly match who was there. And this is very strong, very, so this is called as restriction length polymorphism. So let's look at the activity of this enzyme, BAMH1. So we talked about EcoR1, let's talk about BAMH1. So BAMH1 also acts at a sequence called as GGA-TCC, and from one end to this end, GGA-TTC, and from other end, GGA. So again, this is a palindromic, and this is asymmetrical because they are acting at asymmetrical sites. Although they are acting at the same sequence, but their activity is at a different point. So this is asymmetric. And when this breaks, it gives rise to OH group. This OH group comes from the sugar ribose, deoxyribose, and you have P phosphorus, or from the phosphate group. And then these two can be again covalently conjugated with the help of enzyme called as DNA ligase. So the function of endonuclease and ligase are 180-degree opposite. Means one is a cutter and other is can seal them. One is cutting and other is sort of connecting the digested part. And this has been exploited to insert a DNA. So you can imagine that the plasmid DNA can be cut by restriction enzyme, and that cut can again be sealed with the help of inserting a foreign DNA, which could be insulin gene. So ligase is an enzyme that seals the phosphodiester bond, that catalyzes the phosphodiester bond. This is one of the very old classical plasmid. It's called as PBr322. Also, sometimes scientists, they use alkaline phosphatase to remove the phosphate group to make cloning more efficient. So phosphate group is removed by the help of alkaline phosphatase. And as mentioned earlier, so plasmid is taken, plasmid is cut by restriction enzyme, then the foreign DNA or insulin gene is inserted, and it is the bacteria is transformed. And then, so how do you select bacteria without plasmid and with plasmid, right? So we are interested in bacteria with plasmid, not without plasmid. Why not without plasmid? Because this bacteria will not express insulin. They don't have recombinant plasmid, but this bacteria has. Now, how would we select? Now, plasmids, they also have antibiotic markers. So these antibiotic markers provide resistance. So they have ampicillin resistant gene. So when you take LB agar plate, and in this LB agar plate, you have ampicillin, this bacteria will not grow because they don't have plasmid. If they don't have plasmid, they don't have the ability to resist ampicillin. Where is ampicillin? Ampicillin is in this plate. This is a classical recombinant DNA or transformation experiment. This bacteria will survive happily because they have plasmid, and they have plasmid which has gene for ampicillin resistance. So it will happily grow. But in addition to ampicillin resistant, this plasmid also has insulin. So this bacteria, when growing, while it is making its own protein, it is also making human insulin, right? So I just want to quickly say that students who are, you know, getting into the class with their phone numbers, I am taking attendance. So you need to write in the chat area that, 917 XYZ is your phone number and this is you. So I have to identify each and every student who is attending this class. I have seen where in few meetings where some other people got access and they try to do disrupt the class environment. So in future, if you're entering this class or any class, try to identify yourself. So antibiotics are very important, right? So I told you that bacteria is like lab that provides us tools. What are the tools? Plasmid, restriction enzymes, antibiotics, right? So most of these antibiotics, right? Ampicillin, hygromycin B, kanamycin, tetracycline, they except in ampicillin. Ampicillin is like penicillin. Ampicillin is just like penicillin. It doesn't let the bacteria form the cell wall. So if bacteria cannot form the cell wall, then bacteria cannot survive. That's why penicillin is very effective. But most of these antibiotics, they act at the site of transcription and translation, mainly translational. Mainly, so most of, if you read quickly, they prevent, peptidyl formation or translocation of tRNA, or tRNA, or binds to subunit of ribosome and doesn't let formation of peptide bond, so and so forth. So read this table very carefully. So another topic is DNA hybridization. Now, what is hybridization, right? Hybridization is a technique. So first, let me explain to you what is hybridization. So let's say this is this DNA, double helix DNA, and there is second DNA, right? And these two DNA, they have identical sequence, right? These two DNA A, red color, DNA B, black color, they have same sequence. Now, one thing you remember that if you heat protein, a protein, it will denature. And after denaturing, it will never renature. Renature means it will never come back to its original form. What's the example? Egg. Egg white. The moment you heat an egg white, it becomes, omelette or whatever you say, and it can never come. It is denatured, right? What we eat as sunny side up or omelette, they are denatured proteins, albumin. Similarly is the case with carbohydrate. If you burn the sugar, it will never come back. Lipids, oil, same thing. You heat them, they will not come back. But DNA is something very unique. DNA, you can heat at 95 degrees, at 95 degrees Celsius. So when you heat DNA at 95 degrees Celsius, what will happen? If you heat DNA at 95 degrees Celsius, DNA will separate. DNA will separate 95 degrees Celsius. But the moment you bring the temperature to, let's say, room temperature is only 22 degrees Celsius, or let's say 37 degrees Celsius. The moment you bring the temperature down, this DNA can again form, this DNA can again come back to same thing. So you heat the DNA, they will separate. So how the DNA is separating? The DNA is separating because the hydrogen bond is breaking. The hydrogen bond is breaking. So heat is not good enough. Heat is not good enough to break the phosphodiester bond. So DNA can separate and come back. Now, if you mix these two DNA, right? You take up these two DNA, the red color and black color, and put it in a small tube, microfuge that you see in a molecular biology or cell biology lab. So I'm trying to draw, and you take these both the DNAs, this DNA and this DNA, right? And then you heat this tube at 95 degrees. And then you bring the temperature to 22. So you mix this two DNA, heat this tube at 95 degrees Celsius, cool down this tube. It is possible that you can get a hybrid DNA. So, and how this happened? This happened because both the DNA were complementary, same sequence. So the same sequence, you separated them and they came back. Right? So this is A, this is B. You heat them, you cool them. It is possible that they form hybrid. Why did they form hybrid? Because they had complementarity between them. So they became one. Right? So this is called as hybrid. Now this technique, this simple technique has been used widely. This is also the principle for PCR that we will talk, polymerase chain reaction that we'll talk later. So what happens? Where it is needed, it can be used for many diseases, right? This protocol, this procedure, this philosophy, this concept can be used for disease detection, right? So let's say if there's a child who is born with some kind of a, unfortunate disability, let's say sickle cell anemia, and we want to see whether the child is positive or not. So we can take child's DNA from the blood, and we can have a DNA. We can have a DNA which represent sickle cell anemia, right? And if this DNA from sickle cell anemia, it goes and hybridize to one of these DNA, right? One of these DNAs, then means that the child, the newly born child, is positive. So this is called as, this is hybridization. Right? So first we run the gel, agarose gel electrophoresis, separate the DNA. So what does agarose gel electrophoresis does? Electrophoresis means Electrophoresis means separation of molecules by charge, right? So electrophoresis means separation of separation molecules by charge. So let's say if we have a matrix, right? So let's say if we have a matrix here, and this is connected to a charge, right? So here it is with negative charge, and here it is with positive charge. Now, let's say, so this matrix also has some wells, 2 well, 3 well. This is just in my, so here, in this here, you put DNA. So what is the charge on DNA? The charge on DNA is negative, right? So when you apply electricity, this will move from negative to positive. So what will happen? DNA will come out from this, and they will move on this matrix, and they'll move towards positive, but they will separate out on the basis of size. The larger size DNA will move slowly, and the smaller size DNA will move faster. So electrophoresis is a technique where we separate the molecules by charge. But the mobility is inversely proportional. How do you write inversely proportional? Inversely proportional to the size, meaning that if the size is small, the mobility is fast. If the size is large, so mobility is inversely proportional to the size. So that's electrophoresis. So electrophoresis can be combined with DNA hybridization to exactly detect the DNA of right size. Right? That's DNA hybridization. So what are the important that we need for molecular biology? Yes, bacteria and bacterial products. What are those bacterial products? Plasmid, restriction enzymes, antibiotics, right? And so some of these enzymes are alkaline phosphatase, RNase H, polynucleotide kinase, DNA ligase. So read these list of enzymes. They're very important. So just as a reminder, people who are getting into accessing this class with their phone numbers, they should identify their name. So hybridization, hybrid, right? What is hybrid? Let's say there's a hybrid class. A hybrid class could be that 50% online, 50% face-to-face. So that's a hybrid. So two different things packaged together, two different DNA, but they are complementarity. They are joining by hydrogen bond, hybrid. So let's see. Now, so we were talking about restriction enzymes. Now let's go into the world of RNA, right? And especially mRNA, right? So just briefly, what is the characteristics of mRNA, right? Just briefly. So let's first, mRNA is, for the purpose of simplicity, we draw mRNA as linear molecule, but actually mRNA could have secondary structures, these hairpin loops. So mRNA is that sequence of ribonucleotides that codes for amino acid, genetic code. What is unique here is that on their 5' end, so let's say mRNA could have, 5' end here, and let's say 3' here, this end. At 3' end, this could have poly-A tail mRNA. Here at the 5', they have a capping by methylated G, guanosine. So this is, and they have secondary structure. And triplet nucleotide codes for one amino acid. Now, this mRNA, now in let's say there is a human cell, right? In human cell, this is a cell, and within the cell, there is a nucleus. Within the nucleus, there is DNA, and DNA will transcribe into mRNA or RNA, tRNA or RNA, and that will move to cytoplasm. Now there are 23 chromosomes, or 23 pairs of chromosomes, and there are millions of proteins that are produced in our cells. So what is the link between DNA and protein? The messenger RNA. So at a given point of time in a cell, there is million types of mRNA. But if you're interested in one specific RNA, because in this cell, there are many types of RNA, right? mRNA, tRNA, and rRNA, many types. But you're doing an experiment where you need only mRNA, right? You need only mRNA. So how to take it out, right? So same principle, right? So let's say if you have a needle that you lost a needle and it is present in a haystack, how to find it? So you will need a magnet to take it out, right? How can you find one small needle, right? So the best is to have a magnet that will help you to take out so what is a magnet in case of mRNA? So in what scientists have done, they have synthesized this oligo T, right? Oligo T, and they have labeled this T with a resin. They have labeled this T with a resin which has a weight. Now this particular resin, which is connected to T, they can come and bind to this A by the ability to complementarize. So these T will bind to this 5T will bind to 5A, and then this T is with a resin, which resin means this makes, so this particular RNA, mRNA, becomes attached with a resin. So you can take it in a tube. Right? And centrifuge this tube. And then what will you get in the bottom? You'll get these resins, these red color resins, right? In the tube. And what are these resins represent? That represents mRNA. So you can take out this mRNA. And what will you use this mRNA for? You will use this mRNA to synthesize DNA. Right? Now think what I'm getting at. So here, what is the central dogma of biology? DNA transcribes into RNA, and RNA translates into protein. But there are viruses which can do opposite things. They are RNA virus, and they can make complementary DNA, and then they will again transcribe RNA and protein. And which is that virus that does this? HIV. Which enzyme that can reverse RNA to DNA is called as reverse transcriptase. Now, pardon my spelling mistake, reverse transcriptase. Right? So HIV can reverse this central dogma of biology. Can reverse this central dogma of biology. So which enzyme can convert this RNA into DNA? This enzyme is called as RT. Now this acts as a platform for COVID-19 diagnosis, right? So you could, like I have gone through this. I was not feeling well. I went to the clinic. They took my nasal swab. They must have isolated the RNA. My RNA must have contained COVID RNA, 19 RNA. They reverse transcribed and they did the PCR, and then I was positive. So this, so where this RNA, mRNA could represent, this could represent spike protein, right? So reverse transcriptase is an enzyme that can convert mRNA into RNA into DNA. This DNA is different than the genomic DNA. This DNA, which is produced by RNA, this is called as cDNA. Some people call it complementary DNA, some people call copy DNA. This DNA here is chromosomal DNA. This DNA is chromosomal DNA. This DNA is derived from RNA. So this is called as cDNA, complementary DNA. So this is the backbone for your COVID-19 diagnosis that is going on now. And mRNA again becomes the platform for COVID-19 vaccines. So this is what is given in this slide, that take a cell or tissue, and then extract the RNA, reverse transcription, right? And here in this case, this reverse transcription gives rise to cDNA, and this cDNA can be again engineered into plasmid by restriction enzyme and ligase, and then put into the bacteria. So, and then it can be also used for cloning, right? And we will talk about PCR later, but again, but we'll talk about PCR and RT-PCR later. But let's look at the minute steps of reverse transcription. So what is reverse transcription? Reverse transcription is the process by which mRNA is converted to DNA, which is called as cDNA. And this is through so this is a multi-step process, because RNA is single-stranded, it has secondary structure. So what is written here, what is depicted here as straight line, that's not the native RNA, mRNA. mRNA has secondary structures. So to make mRNA linear, we have to heat the mRNA at 70 degrees Celsius. Once you heat the mRNA, then these mRNA become linear. And then we add reverse transcriptase. But the tragedy with reverse transcriptase is that enzyme that is derived from retrovirus, such as avian myeloblastosis virus or myeloid leukemia, AMV or MLV. So reverse transcriptase, they need a primer. They need a pre-existing oligonucleotide to start synthesis. So what is our goal of experiment? We get the mRNA, and we want this mRNA to convert to cDNA. For converting into cDNA, first we need reverse transcriptase, and that can be available commercially for, let's say, AMV or MMLV. Now, what we do here, we need to synthesize the hybrid DNA, right? Hybrid to this mRNA. So we need the building blocks, DATP, TTP, GTP, CTP. And we need a cofactor, magnesium. With all these, we also need oligo DT, that I showed you. And this oligo DT binds to the 3' of mRNA. And then this synthesizes the cDNA. This cDNA is hybrid of mRNA and cDNA. So newly synthesized, this wavy structure is complementary DNA. This hybridizes to mRNA, messenger RNA. So this has to be broken. So how to do it? So let's go to the next slide. So this is mRNA. We need oligo DT primer for the enzyme to work. Then a complementary cDNA is synthesized. Then this RNA is removed by an enzyme called as RNase H. I'll come back and write it. And then only one strand of cDNA remains. So again, you need a second enzyme, third enzyme, DNA polymerase 1, to start the synthesis of second strand. And this DNA strand is like hairpin. So you need the fourth enzyme, S1 nuclease, to generate. So how many enzymes you need for reverse transcription? One, reverse transcriptase from AMB or MMLV viruses, because retroviruses, they have enzyme called as reverse transcriptase. Once reverse transcription is done, you need to remove mRNA cDNA hybrid through an enzyme called as RNase H. Then you only have one strand of cDNA, and this cDNA, to convert into double-stranded DNA, you need DNA polymerase. Now you get DNA polymerase, you get DNA double-stranded, but you have this connected here. So this has to be removed. So this is removed by S1 nuclease. So to summarize this, let me make it simple. For the process of reverse transcription, so number one, extraction of RNA. Right? Then how do you get to mRNA? To get to mRNA, you need oligo DT. And then you need reverse transcriptase as first enzyme. This enzyme produces hybrid of mRNA and cDNA. Then you want to remove the mRNA because you are interested only in cDNA. So you need another enzyme called as RNase H. It removes the mRNA. But when it removes the mRNA, the cDNA is single-stranded. Then you need the third enzyme called as DNA polymerase 1 for the second strand synthesis. Then I told you that this strand that is formed is like this. So you need to break this loop here. This enzyme, this is broken by another enzyme called as S1 nuclease. So let's say if I give you various situations and I say make the right sequence of enzymes that work in reverse transcription. So first will be reverse transcriptase, second RNase H, third DNA polymerase, and fourth S1 nuclease. Right? So that's reverse transcriptase. Now, so what we discussed here I'm sorry, I didn't realize that my camera is off. So now look at this chapter. This is quite a decently big chapter with a lot of information: history, structure of DNA, mRNA, then restriction enzymes, bacteria, plasmid, types of restriction enzymes. Now, so now is that how do we do the uh, cloning, right? Now, what is happening that as we are the technology is improving day by day. So molecular biology classically started after the discovery of DNA 1953, but to put things in an efficient manner, it really got a momentum during late 80s and 90s, when these plasmids were purified, digested, and for cloning. But we reached a stage where we needed a plasmid that can incorporate large sequence of DNA. Because think about human sequencing, right? Sequencing of human genome, that's quite a meter of DNA, and how to put them into plasmid. So you need a larger category of plasmids that can incorporate large amount of DNA. So this is the last slide. These are various kinds of vectors. Again, vectors are those agents that carry parasitic agents like malaria or leishmaniasis. But here, when we talk of vector system, these are the DNA that can carry foreign DNAs. For example, plasmid. Plasmid can be found in E. coli. And the capacity to plasmid has a capacity to insert until 10 kb. Beyond that, plasmid will not work. Bacteriophage that infect E. coli, it can carry plasmid from 10 kb to 20 kb. 1 kb is equal to 1000. So 10 kb is 10,000, 20 kb. So bacteriophage is a better vector than plasmid. Cosmid is the hybrid of bacteriophage and plasmid. So they can carry 35,000 to 40,000. Then, East artificial chromosome, they can carry 100,000 to 200, 100,000 to I believe this is 2 million size nucleotides of DNA. So, these are, vectors are the tools that can carry foreign DNA, and they can carry foreign gene to express proteins that can be used for commercial purposes to treat diseases, so on and so forth. So with this, we finish this chapter 1. And because I changed the date on the Blackboard also, the submission of assignment is next Tuesday. It was initial deadline was today, but because I believe last Tuesday was Friday, so I've extended the date. And your first quiz is on February 21st, because everything moved up by one date. I got a question in the chat area. Can you tell me what's the question is? I'm not able to see the chat. Yeah, maybe now I can see. Okay. So just let me have a look. Yeah, who is 646 phone number? Please tell me. 646-353-6554. May I know who the guest is? Then there is somebody with 917-412-9826.
Sandra Dyer Morrison.
Say again?
Sandra Dyer Morrison.
Sandra, okay, great.
And Professor, I was with you last Thursday as well, but I did not know how to operate the phone.
Thanks for telling me. And who is 929-530-9680? May I know who the student is with phone number 929-530-9680? Yeah, Miss Redji, I got your attendance. Are there any questions?
You get my name, Professor Bernard?
Tell me the full name.
Oh, Bernard Sajin.
Yes, I got you.
All right, thank you.
Most of students I got as they came into the class, but later when I started teaching, I could not focus. I can only focus on one thing. But still, I cannot find people who.
What about me, Professor?
Yeah, Rimsha, I got you. So I still don't know who is with number 929-530-9680. So if you don't reveal, then I will not let you in my class next time. And are there any questions? If there are no questions, I believe.
What is the first quiz, sir?
Hello?
What is the first quiz?
21st of February. Only chapter 1, multiple choice questions.
Okay, thank you.
Any other questions? If not, then I'm ending this class. Have a good rest of the day or evening. See you. Yeah, Miss Redji, I announced that I have marked you present.
Okay, I missed that. Thank you so much.
Bye.